So I'm really pleased to be hosting today Dr. Danny Xie and Dr. Bernard Choi to discuss the recent PLOS ONE Lab Protocol paper entitled Semi-Automated Protocol to Quantify and Characterize Fluorescent Three-Dimensional Vascular Images and the associated detailed protocol in Protocols.io. Dr. Choi is a professor of biomedical engineering and surgery at the University of California, Irvine, and the interim director of the Beckman Laser Institute and Medical Clinic, a multidisciplinary research center dedicated to the translation of optical approaches from the benchtop to the clinic. He received his bachelor's in science degree in biomedical engineering from Northwestern University and his master's and PhD degrees in biomedical engineering from the University of Texas at Austin. Professor Choi's group is focused on developing technologies and methods to study microvascular structure and function, with current applications in neurology, critical care, and urology. Dr. Danny Xie is a former graduate student in biomedical engineering at the University of California, Irvine, where he received his master's and PhD degrees in biomedical engineering. He received his bachelor's in science degree in biomedical engineering from the University of Florida. His PhD work focused on imaging of microvasculature using tissue clearing and developing methods for quantification of microvasculature. Danny, Bernard, thank you so much for being here with us.
Our pleasure.
Can either of you briefly explain what the protocol is about, how to use it, and why it is relevant?
So I can go off and start. So our protocol is largely focused on the vascular network, specifically for visualization of the vascular network and quantification of the vascular network. Some of the reasons we are interested in studying the vascular network is because the blood vessels are very essential to the health and function of different tissues, as it provides nutrients as well as removes waste products. So overall, this protocol is focused on trying to utilize a specific vascular label to help image the vasculature, as well as incorporating tissue clearing to enable us to create transparent tissue and collect three-dimensional images of the vascular network, as well as quantification of those images. So we hope to put together a protocol that was simple and reproducible for different researchers to hopefully be able to help facilitate research in this field.
And to better appreciate the significance of the protocol, can you help us understand what was the state of the art? In other words, what was the motivation to optimize the protocol? And if anything similar existed in the literature, why those protocols didn't work for you?
So definitely there are existing methods for labeling the vasculature, as well as different combinations of methods for clearing tissue, as well as different methods for quantification of vascular images. There's some pros and cons to different methods, and sometimes they're not necessarily compatible with all of these different steps. So one thing we wanted to do was kind of put together a protocol of what we've been doing that does put together a lot of these steps together, whereas many other publications might just focus on one area versus the others.
So you compile everything in a single workflow.
Mm-hmm.
And going deeper into the protocol itself, for the new user, what are the most critical steps in this workflow and protocol?
Yeah. So I guess Yeah, so in terms of the workflow itself, the obviously getting the vascular label into the bloodstream is it's a critical step with this, and I think a lot of people use it with mouse models. A lot of people use tail vein injection in order to get different dyes, different agents into the vasculature, and it's a tricky technique. For some people that when they get very good at it, they almost become invaluable to a lot of those projects. We found that the retroorbital route, it looks a little bit strange to people who haven't seen it before, but it's basically injecting into this pocket that's right between, is basically right next to the eye. So you basically will just put a needle right into that area, and then you inject the dye directly into there. It gets into the bloodstream quite easily, and it's very when the procedure doesn't work, it's also very obvious as well. So there's very few, I think, false positives in terms of the injection. And once you do a few of them, it's, I think, a very simple injection route to get this dye into the bloodstream. So that's one of the big things. And Danny, I'll leave the other steps to you.
So for us, we are primarily come from an engineering background, so a lot of these animals procedures we're less familiar with, and definitely for newer individuals who are using this protocol, if they're less familiar with a lot of the animal steps, including the injection, those can be tricky to get the hang of, and it's good to have people nearby that are more familiar with the animal work. In addition to that, if you are interested in using additional labels with your samples, it's important to make sure that they are compatible in terms of the different procedures, making sure that the different labels are able to stay in the samples, and also making sure that if you are using fluorescence, that the signals don't overlap, so you can clearly distinguish between the different labels that you have.
And as you were developing the protocol, was there anything that you tried but didn't work and didn't make it to the final version of the protocol?
So we did use different vascular labels in the past. One of the dyes that we used before is known as DII, which is a lipophilic dye. One of the main issues we had with that vascular label is it was difficult to get it to label some of the smaller vessels in the tissues, which was very important for our research. So we ended up moving away from that and using the dye known as lectin, which is a glycoprotein that is able to attach to different glycoproteins within the endothelial walls of blood vessels, and we found that that was a much better vascular label. In addition to that, we also used a different clearing approach before, using a commercially available product, and we had some issues with the quality of the clearing, so we also transitioned to a different clearing procedure after that as well.
Yeah, one of the things.
Sorry, go ahead.
Oh, sorry, Gabriel. One of the things with the dye, switching from DII to lectin is the administration route it's different. So DII, you inject, it's an intracardiac injection, and so it then relies on the quality of the injection as well as the rate at which you inject the dye into the bloodstream to be able to circulate properly. And this is, as Danny had mentioned before, why sometimes it's difficult to label smaller vessels with this technique. Whereas with the lectin, when you inject it in vivo with this retroorbital route that I just spoke about, you're then using the heart to naturally circulate the dye through the bloodstream. So that we think is a pretty big advantage of the using the lectin over in this case, the DII or any other labeling agent that you would do intracardiac.
And the paper, if I recall correctly, the paper that you published in PLOS ONE focuses particularly on cardiac tissue in mice. Have you tried, and correct me if I'm wrong, but have you tried to extend the clearing procedure to other tissues and to other model organisms?
In terms of different tissues, we have had data for looking at different organs, including the mammary gland, skin, and the brain is a large focus of what we currently do. So overall, with this method for labeling of the vasculature, it labels all the blood vessels within the mice. So we're able to take any of these organs and process them in a similar fashion and obtain images of them. In terms of other organisms, we have not done anything outside of mice right now, but the procedure should be able to be transferred over to different organisms, including rats or any other rodents. Some of the procedures will probably have to be modified for the larger animal, but it should be able to work for other animals.
No, I guess one thing I do want to be clear about is the clearing technique that we do use. It's one that's it's not one that's been developed by us. It's one that we've adapted for this specific procedure, and we made small modifications to it that so it does build on a body of work that others have done in terms of using this clearing protocol for the brain and for other organs as well. So there's slowly but surely becoming a body of I guess different organs that have been that have been cleared with this procedure. The combination with the vascular label is something that's maybe a little bit more unique to, I think, to the work that we've been doing in the lab.
And I would like us to change gears a little bit now and talk about the motivations. First, to make the protocol fully available publicly, and the second, to get it peer-reviewed as a standalone methodological paper.
So one of the reasons we wanted to publish it as protocol was oftentimes when we're looking at certain manuscripts and we're interested in trying to replicate some of their procedures or adapt for our purposes, we often find that there might be like some small minor details that are either left out or maybe not explained fully. And those details tend to be very routine for individuals who are familiar with the field, but for any individuals that are outside of the field trying to kind of get their feet wet for the first time, those details are hard to figure out on their own. So that's one of the reasons we wanted to publish this as a lab protocol. In addition to that, we did also have a lot of our collaborators who are interested in sort of the procedures that we do, and we wanted to put together a sort of cookbook style protocol that they could potentially follow as well.
Yeah, so the genesis of it really was it really did start off as some of our collaborators would ask how do you do that, and so Danny with assistance from some undergraduates in the laboratory were able to put together a really nice initial protocol that it was there were some edits made to it between when we originally developed that protocol, for internal use, and then what's now available on the protocols.io website. But to a large extent, it's many parts of it are pretty identical. So the genesis of it was already, it was already initiated, and then the PLOS ONE publication plus the protocols.io gives a really nice kind of forum to be able to share it.
And can you share with us how, and if and how, the feedback that you received from the peer reviewers at PLOS ONE shaped the final version of the protocol?
So one feedback that was requested was to kind of expand some of the validation that we did, mainly just including like expanding the number of kind of test images we were using to validate. So we kind of went back and put more time into that step of the protocol.
And the last question that I have is I know this is a relatively young paper and protocol, so it might be a little bit speculative at this time, but how do you see, or what do you think the impact of publishing the protocol and the paper will be for each of your careers at different stages, right? An established professor and a young scientist.
So I can answer that first. So a large focus of my dissertation ended up revolving around the different parts of the methodology that we included in this protocol. In addition to that, some of the projects that I worked on kind of spun off of applying these techniques to work with some of our collaborators, looking at neurology, biomaterials, and cancer biology. So it was a pretty fundamental part of my PhD work.
Yeah, I could say, I guess from my view, or from my experience, this concept of tissue clearing, at least to start with, is something that I learned about in graduate school, and I was fortunate to work with one of the pioneers in this field. It was for skin, and it was completely different chemical agents and for completely different purposes. And so it's a field that, since graduate school, it's something that we've been working on, just once again, specifically for the skin to start with, but then through basically some just serendipity, and also just through some of the urgent needs that some of the needs that we had for different projects, we evolved from the skin over to ultimately to the brain and to other organs. And so that really has allowed, as Danny says, really allowed for it's really engendered excitement, I think, in with some of the our colleagues at UCI, and also from other locations as well. And then it's just kind of naturally turned into this approach that we're currently using. And we think it's because a lot of it because of by spelling out all the details, and then also I think with the simplicity of the image analysis approach in particular is something that's relatively easy for most people to code in whatever language they want to. I think it really allows for relatively easy adoption of these techniques to try to do really quantitative analysis of a fairly complicated structure, which is in our case we're mainly focused on the brain, but you look at the vascular networks and all these other organs, and there's a lot of beauty to the vascular networks, and there's a lot of there's a lot of complexity, and there's a lot of need actually to be able to study the vascular networks and how they how they evolve with age by itself, or how they evolve with disease. And so I think from that perspective, I think it really does open up a lot of possible ties to anybody that's interested in looking at the vasculature for whatever field of study they're interested in.
Well, thank you so much, Bernard and Danny. I don't have any other questions. I don't know if there's anything that you would like to mention that wasn't touched in this conversation.
I think we covered most things. Yeah, I can't think of anything. I guess the only other thing would be, if anybody is interested in learning more, they're obviously more than welcome to reach out to Danny and to me. At any point, we're happy to share what we've learned, and we're happy to to help out where we can.
Thank you both so much.
Thank you.
Thank you, Gabriel. This was fun.